Good day, students. You're welcome to the third topic in the series of CRP 305, crop breeding and genetics, and I still remain your facilitator, Alaudi Yetunde. Last week, we talked about Mendel's principles and its laws. So this week, we'll be basically illustrating most of those principles and laws. So we'll be illustrating on monohybrid crosses, on dihybrid crosses, as well as trihybrid crosses. Also, we are going to be talking about the tool in investigating the goodness of fit, which is the chi-square. So it involves more of calculations this week. And trust me, if I say calculations, I don't mean any form of lengthy algebra. It is simple and straight to the point. So take your pen and your paper and let's be on our jolly ride. Thank you and stay blessed. Like I said in the previous slide, my name has not changed. I'm still your facilitator, Alaudi Yetunde, and my email is on the screen. We will be talking more about the Mendel's laws and the type of crosses that we have. The first cross we'll be talking about here is the monohybrid cross, and it simply means a cross in which a single pair of alleles is considered. So where a single pair of alleles is considered, we call it a monohybrid cross. So monohybrid crosses involve one gene with two alleles, like you can see in this picture here. This is a cross between a pea that is homozygous for yellow seeds. You can see the homozygous sign. These are the alleles that are representing the yellow seeds. And the pea plant that is homozygous for green, you can see it in lowercase y. And they will produce heterozygous offspring. This is what the heterozygous looks like. So if you want to write it out, these are the alleles that are produced from the homozygous dominant gene, and these are the alleles that are produced from the homozygous recessive. So when you combine it this way, then you get your heterozygous, which is 4/4. So that is it for the monohybrid cross. A good example is here also. A pair of genes govern color in seed. The dominant allele B governs the seed color, the black seed, and the recessive produces the white. So the parental generation is symbolized by P that is written here, and the first filial or the first generation is symbolized by F1. So you can see it here, a cross between the homozygous black, which is the capital B, the alleles are capital B, and the homozygous white, the alleles are small letter B and small letter B. So these are the gametes that are produced, and when they combine it, you get the F1, which is black, but it is heterozygous black. Moving on to the dihybrid crosses, this involves two genes. So when there are two genes, that means there are four alleles. So the dihybrid is going further, a step further, more than the monohybrid cross, and it involves two genes and four alleles. And an example here is crossing a pea with yellow round seeds with another variety with green wrinkled seeds. You can see that two genes are involved, the yellow and the round seeds, and then the green and wrinkled seeds. So let's put that aside. You can also see here in this picture here, this is an example of a dihybrid cross. Here the father is heterozygous for a trait, and you can see it represented by capital A and small letter A. And you have the homozygous dominant like this, the heterozygous like this, and the homozygous recessive like this. So when you have a dihybrid cross, these are the alleles that are produced. Okay, These are the alleles that are produced. This is a dominant trait. This is heterozygous dominant. This is heterozygous dominant also, and this is recessive, homozygous recessive. So you can see, to make it easier for you, you can align the gametes this way, vertically and horizontally, and then you cross them. You cross them across. When you get A, capital A and B, and capital A and B, you get your resultant cross here in the squares that are provided here. So at the end of the day, if you count the dominant gene, the ones that have any capital letter represented, then you have, this is 1, 2, 3, 4, 5, 6, 7, and 8. And you have the other one, which is dominant recessive. You have it here. If you count, this is 1, this is another one, and this is another one. So at the end of the day, you're going to get a dominant, which is 9 out of 16, and a recessive, which is 3 out of 16. So that is it about the dihybrid cross. Example is here also. These are the parents, the capital A, capital B by small a and small b, and these are the gametes that are produced. So when you combine them, then you get an F1 like this, yellow and round, heterozygous for yellow and round. And to make things easier, if you want to know the number of gametes that are formed from any kind of cross, this is the formula you use, 2 raised to power n, where n is the number of gametes. So, and for the number of genotypes, you use this formula, 3 raised to power n. So that is 4, and the number of genotypes that will be produced is 3 raised to power 2, that is 9 genotypes. So it's important to know that in a dihybrid cross, the phenotypic ratio, that's what you see, is ratio 9 to 3 to 3 to 1. So that is the classical Mendelian ratio for the dihybrid cross. So I have a short class activity for you here, and what I want you to do is to self the product from this cross. Okay, you self the product from this cross. This is heterozygous. This is one allele, this is another allele, and this is another allele. So when you're selfing it, I want to know if you understand it. So how many gametes do we expect from this cross, and how many genotypes can we get? So I need you to use the Punnett square to get the expected genotypes and the ratio. So still continuing with the dihybrid cross, we want to know the phenotypic and the genotypic ratio. And when you perform a dihybrid cross, at the end of the day, you get the ratio 9 to 3 to 3 to 1. And in this case, you have the yellow round seeds, which are 9, the yellow wrinkled 3, the green round 3, and the green wrinkled, which is the recessive, just 1. So that's the classical Mendelian ratio for a dihybrid cross. Further, we are talking of trihybrid cross, and tri means 3. So 3 pairs of contrasting traits are involved in this particular kind of cross. 3 pairs of contrasting traits are involved, and an example is that if you consider a cross between these, you can see this allele, capital A, capital A, another allele, capital B, and another allele, capital C, capital C. So 3 pairs of contrasting traits. Okay, this may be for tallness, this may be for flower color, this may be for seed color, contrasting traits. And individuals of this, when you now cross them, this is dominant for this trait, and this is recessive for this trait. So all the F1 individuals are heterozygous. You get something like this for the F1, the capital A small letter, in a mixed form. So wherever you see capital, that's the dominant, capital A, B, or C, then it's dominant for that particular kind of trait. And in this chart here, you can see this is another example. We have tall, pinch pod, and axial plant is represented by these alleles, and then you have the dwarf, inflated, and terminal plant represented with these alleles. So the F1 is all tall, inflated, and axial. Okay, that's the dominant. Tall, inflated. This is for axial, and this is for inflated. So this is what you get. This is a trihybrid cross. Over here, this is just illustrating more. So in a trihybrid cross, you have the number of gametes that are formed with the formula 2 raised to power n. You know the number of gametes that are formed, and with the formula 2 raised to power 3 raised to power n, you know the number of genotypes that are formed. In this case, there are 1, 2, 3, 4, 5, 6, 7, 8 gametes that are formed. Okay, so when you put them horizontally and vertically, you can cross them to get these alleles. Okay, these alleles, and these alleles are 1, 2, 3, 4, 5, 6, 7, 8 by 8. That is 64 in number. In a trihybrid cross, you have 64 different gametes that are produced. So you can see this is dominant. This is also dominant. Wherever you see the capital letter is dominant for that particular trait. But when you are coming down here, you can see that is recessive. You see, recessive for this trait, recessive for this trait. So there is a way that the gametes are distributed across the crosses. So this is what you have in a classical Mendelian ratio. So this is just further illustrating that this is what the parents look like. 3 different alleles, and the gametes produced are this, and this is what the F1 will be like. Now when you self the F1, you get 3 different gametes. So you have n to the 3, sorry, and you have 8 gametes. So when you place 8 by 8, you get 64 genotypes. Okay. Now there's a table here that will show you what to expect when you perform your crosses. Now here the first column is the number of heterozygous gene pair. The second column is the number of different gametes. Third is number of different F2 genotypes produced. And the fourth one here is the number of different F2 phenotypes that are produced. These are the formula that you will use, the formulae that you will use for them. This is N, the number of gene pair. For the gametes, you have 2N, genotypes 3N, and phenotypes 2N. So if you have only one gene pair, you have 2 gametes produced, 3 genotypes produced, and 2 phenotypes produced. If it is 2 gene pairs, you have 4 gametes, 9 genotypes, and 4 phenotypes. If it is 3, you have 8 gametes, 27 genotypes, and 8 phenotypes. If it is 4, you have 16 gametes, 81 genotypes, and 16 phenotypes. While if it is 10, you have 124 gametes, 59,049 genotypes, and 124 phenotypes. So this will just give you an idea of what to expect when you perform your crosses. So chi-square now, we want to know what chi-square means, and it's just a tool. It's a test tool that helps the investigator to determine goodness of fit. You want to know how well that model fits into what you're doing, and it's important in genetics to be able to evaluate this observed deviation, because sometimes you do something on paper, and when you get to the field you see something that is slightly different. It doesn't have to be all the way deviated from what is on paper, but it just helps you to be in check so that you are not doing something that is ambiguous. Okay, so that is what the chi-square is for, and it is a statistical tool. And here you evaluate how well the observed data fits into the expected ratios. You know what to expect from Mendelian ratio, and then you go to the field and perform your experiment. So what you get is not, it may not be exactly what is supposed to be in the Mendelian ratio, but it shouldn't be totally different from it. So that's what they're saying. Like in the monohybrid cross, the phenotypic ratio that is expected in the F2 population is a ratio 3:1. And in the dihybrid cross, like we talked about earlier, the phenotypic ratio that is expected is 9:3 to 3:1. So we're going to use, you have your um observed to be 740 minus 750, okay, and that's observed minus expected squared all over expected, if you remember what the formula is. So that is what we did here, and you get minus 10. You don't have to worry about the minus, because when you multiply it, when you square it, the minus becomes plus. So at the end of the day you get 0.13. So the chi-square value is 0.13. Got for your calculated. And for the white cotyledon in ratio 1, the expected value observed minus expected all over squared all over observed, you get 0.4. So a summation of this chi-square, both the calculated, both the yellow and the white cotyledon, is 0.13 plus 0.4, which gives 0.53. Therefore, the calculated chi-square value is 0.53. And from the table, because there's always a table like your four-figure table, you have the chi-square table. That is where you can check the tabulated value. So you check the tabulated value with a degree of freedom of 1, because the ratio here is 3:1. So that is 2 ratio. So what you do is you subtract 2 from 1 to get 1, and the degree of freedom is 1. At a level of 0.05, on the table what is there is 3.83, 84, I beg your pardon. The tabulated value is 3.84, while the calculated value is 0.53. So in this case now, the tabulated is greater than the calculated, so it means it is not significant, and it could as well, that what that means is that it could fit perfectly into the Mendelian ratio of 3:1. So I hope that is explanatory enough. Now you have a class activity here, and I just expect you to use the same method that I used. A breeder made a dihybrid cross and is expected to see a phenotypic ratio in 9:3 to 3:1. That is the Mendelian ratio for a dihybrid cross. but cannot he could not see the expected ratio. Then he suspected that there was linkage in the chromosome. So the man planted 800 plants, and this is his observation: 440 to be yellow and 160 to be um 440 to be yellow and round, and 160 plants to be yellow and wrinkled, and you have 130 plants to be green and round, then 70 plants to be green and wrinkled. So the question now is, what did the breeder expect to see? So I want, in the next class, we will work it out, and I will show you what to do, and expect you to work it out on your own, so that you'll be able to understand better. Question is here: a breeder made a monohybrid cross, and he expected to see a phenotypic ratio of 3 to 1, yeah, which is the Mendelian ratio. But he observed, this is the observed value now, 215 plants to be resistant and 85 plants to be susceptible out of 300 plants that he planted. So the question now is, use chi-square to prove whether what is observed, what he observed is what he expected to see. And I want you to draw your hypothesis and a valid conclusion. So I'll give you some time to do that. Having said that, I want you to work on those calculations, and in the next class we will meet and deliberate on your answers. Having said that, I want to thank you for a nice lecture, and I hope that you understand better. If you have any questions, you can always chat me up or send me an email, and I wish you all the best. Thank you, and God bless.